Welcome to the deep dive. Today, we're going on a deep dive into the world of Borrelia species, and specifically with you in mind, a medical microbiologist. We'll be using the book Principles and Practice of Clinical Bacteriology, second edition by Gillespie and Hockey, and hopefully we can give you some interesting facts and clinical insights.
Yeah, sounds like it'll be a really fascinating deep dive. Borrelia are pretty unique bacteria, and there's a lot to unpack.
Definitely. So why don't we just jump right in? First things first, what exactly are Borrelia?
Well, you can think of them as tiny spiral-shaped bacteria, but here's where things get interesting. They're not your average everyday bacteria. You see, Borrelia have these unique flagella, internal ones, that basically act like tiny propellers wrapped around their bodies. This setup makes them incredibly motile, which allows them to navigate through really thick environments, like blood and tissue. But here's the kicker: despite their ability to thrive in these challenging environments, they're surprisingly delicate. Sensitive to things like UV light and drying out.
That's kind of surprising.
Well,
You wouldn't think a bug that lives in ticks would be so sensitive.
right. It's a real paradox, isn't it? It really highlights how well adapted they are to their unique life cycle. They have to survive in both ticks and mammals.
And speaking of different hosts, our source mentions a whole bunch of different Borrelia species. Which ones should we, as medical microbiologists, be most familiar with?
Well, out of all the Borrelia species out there, the ones you're most likely to encounter are Borrelia burgdorferi, Borrelia guerini, and Borrelia afzelii. These are the main players responsible for Lyme disease, and they're all spread by those pesky Ixodes ticks.
Ah, Lyme disease, the infamous tick-borne illness. It's interesting that you mentioned Borrelia's unique flagella. How does that structure actually play a role in causing disease?
That's a great question. Those internal flagella aren't just there for looks. They actually contribute to the bacteria's ability to cause disease. You see, the motility that these flagella provide allows Borrelia to spread really quickly from the tick bite, disseminating throughout the body and even crossing the blood-brain barrier. It's this ability to move quickly and get into tissues that helps explain why Lyme disease can have such a wide range of symptoms, affecting all sorts of different organ systems.
So it's not just about the bacteria being there, it's about their ability to move around and invade different tissues. Makes sense.
Exactly. And to make things even trickier, these bacteria are masters of disguise. Their outer surface proteins, particularly a group called OspA-F, actually change depending on where they are, like whether they're in a tick's gut or a human host.
Wait, hold on. They can change their outer appearance. What's that all about?
It's pretty amazing. It's like they're wearing different disguises to evade the immune system. When they're in a tick, they express certain proteins that help them survive there. But as soon as they're transmitted to a human, they switch things up. They start expressing different proteins that can help them avoid detection by our immune system.
Sneaky. I'm starting to see why Lyme disease can be so difficult to diagnose and treat.
Exactly. And their trickery doesn't end there. Not all Borrelia are created equal. Different species and even different strains within a species can have varying levels of infectivity, and they can cause different levels of severity in the disease. This all comes down to differences in their plasmids, which are like little bonus DNA packages that give them extra abilities. Think of it like a superpower toolkit. Some have more tools, some have less, which makes some strains more dangerous than others.
So it's not just about whether you get infected with Borrelia, but which specific strain you encounter.
Yeah.
It's a really important piece of the puzzle for us in the lab.
Absolutely. And there's this one protein in particular, OspC, that has caught our attention. It's highly variable, and get this, it can actually be used to predict how invasive a particular strain is likely to be. By analyzing the OspC protein, we can get a sense of whether a strain is more likely to stay localized or if it's going to spread rapidly.
So for us, as medical microbiologists, knowing about OspC variations could really help us assess the potential severity of a Lyme disease case and make better treatment decisions. That's huge.
Precisely. It's almost like having a crystal ball.
Yeah.
It gives us a glimpse into the potential future course of the disease.
Wow, that's really incredible. Okay, enough about these bacteria themselves. Let's talk about what we see in patients. What are the hallmark clinical features of Lyme disease?
Well, the classic sign is the erythema migrans rash, or EM. It's that distinctive bullseye rash, but it can also present in a lot of other ways. Sometimes it's a solid red rash, sometimes it has multiple rings, and sometimes it can even be completely absent.
So we can't always rely on that textbook bullseye rash for diagnosis. What other symptoms should we be looking for?
Well, along with the rash, early symptoms can include things like fever, fatigue, headache, muscle, and joint aches, and swollen lymph nodes. Basically, flu-like symptoms. These symptoms are pretty general, which is why it's super important to consider Lyme disease as a possibility, especially in areas where Ixodes ticks are common. and particularly during peak tick season.
It's like a clinical puzzle, isn't it? Trying to
Yes.
piece together all the clues and figure out if Lyme disease is the culprit.
Exactly, and it's a puzzle we need to solve quickly, because if Lyme disease is left untreated, things can get a lot worse.
That's a good point. So let's talk about the potential consequences of untreated Lyme disease. What kind of risks are we talking about?
Untreated Lyme disease can have some pretty serious long-term consequences. It can spread to the heart, causing carditis, which can lead to arrhythmias and even heart failure. It can also affect the joints, causing Lyme arthritis, which leads to persistent pain, swelling, and stiffness. And in some cases, it can even invade the nervous system, leading to neurological problems like meningitis, facial palsy, and nerve pain.
Wow, that's a serious reminder that early diagnosis and treatment are crucial. We really need to be on top of this.
I completely agree. Recognizing the signs and symptoms early on can make a huge difference in how these patients do in the long run.
Okay, so let's say we suspect Lyme disease based on the clinical presentation. How do we actually confirm the diagnosis in the lab? What tools do we have as medical microbiologists?
Well, microscopy can sometimes be helpful, but those tiny Borrelia are pretty hard to find. They can be tough to spot under a regular microscope, especially because they like to hide out in tissues.
So microscopy alone isn't always reliable. What's considered the gold standard for diagnosis?
Culturing the bacteria, isolating and identifying the specific Borrelia species is the most definitive way to diagnose Lyme disease. But it can be a bit of a pain. Borrelia are kind of notorious for being finicky growers, and it can take a long time to culture them.
And sometimes we just don't have a lot of time, especially when we're trying to make treatment decisions.
You're right, so that's where serological tests come in. These tests look for antibodies in the blood that are specific to Borrelia. The most commonly used are ELISA and Western blot. ELISA is usually the first step, and if it's positive or even borderline, we'll confirm with a Western blot.
I've always found those serological tests for Lyme disease to be a little bit tricky. What are your thoughts on false positives with ELISA and when you decide to order a Western blot?
That's a great question. You definitely have to be careful with interpretation. False positives can happen, especially early in the infection when antibody levels are still low. So I always consider the patient's clinical presentation, their exposure history, and any other relevant findings before I decide whether to order a Western blot. It's really about putting all the pieces of the puzzle together.
It really does sound like detective work. Are there any other diagnostic tools we can use to help crack the case?
We do have some newer techniques that can be helpful in certain situations. Like PCR, for example, can detect Borrelia DNA in different samples, which can be useful when serological tests aren't clear. And then there's the Lyme borreliocidal antibody test, or BAT, which looks at how well the patient's antibodies can kill the bacteria. This can give us additional information to support the diagnosis, especially in those tougher cases.
It's amazing how far we've come with diagnostic technology. It's like having a whole arsenal at our disposal to fight these microbial invaders. Speaking of the bigger picture, let's talk about the epidemiology of Lyme disease. What are some of the key trends and patterns that we're seeing these days?
Lyme disease is actually the most common vector-borne illness in the United States. It's mainly concentrated in the Northeast and Midwest, which reflects where those Ixodes ticks are found. And unfortunately, it's becoming an even bigger problem.
It's concerning. What factors are contributing to this rise in Lyme disease cases?
Well, there are a few things happening. One is the expansion of tick habitats due to things like deforestation and suburban sprawl, which brings people into closer contact with tick populations. Climate change is also playing a role because warmer temperatures create more favorable conditions for ticks to survive and reproduce. And then there's the deer population. Deer are the main hosts for adult ticks, and their increasing numbers in many areas are contributing to a larger number of ticks overall.
So it's this complex mix of environmental, ecological, and human factors that are driving the spread of Lyme disease. What can we do to protect ourselves and our patients from these tick-borne threats?
Prevention is always the best medicine. So educating patients about the risks, encouraging them to avoid tick-infested areas, wear protective clothing when they're outdoors, and use insect repellents can really make a difference.
Those are all great strategies for individuals. But what about larger-scale efforts to control the tick populations themselves? What's being done on that front?
There are ongoing efforts to control tick populations through habitat modification and the use of acaricides, which are chemicals that kill ticks. However, these approaches can be challenging and sometimes controversial. Habitat modification can be tough to implement on a large scale, and the use of acaricides raises concerns about their environmental impact and the potential harm to other organisms.
I see. So it's a real balancing act, trying to find effective and sustainable ways to manage these tick populations. What about a vaccine? I thought there was a Lyme disease vaccine at one point.
There was actually a vaccine called Limerix, which was based on the OspA protein, was available for a while.
Yeah.
It was shown to be pretty effective at preventing Lyme disease, but it was withdrawn from the market back in 2002 due to low sales and some unfounded concerns about potential side effects.
Oh, that's unfortunate. A vaccine could be a real game changer in the fight against Lyme disease.
Yeah.
What about prophylactic antibiotics after a tick bite? Is that something you recommend?
That's a tough one. There's actually some debate surrounding the use of prophylactic antibiotics. The decision really needs to be made on a case-by-case basis, weighing the risks and benefits. Factors like the type of tick, how long it was attached, and the patient's individual risk factors all come into play.
So there's not a one-size-fits-all approach. It really requires looking at each individual situation.
Exactly. There's no easy answer. But with a careful assessment and shared decision-making with the patient, we can make the most informed choice.
All right, let's move on to treatment. What are our go-to antibiotics for Lyme disease?
Early treatment is key for Lyme disease, and the good news is we have a number of effective antibiotics we can use. Doxycycline and amoxicillin are usually the first-line choices for those early-stage infections, and they work really well in most cases.
And what about those more complicated cases where the disease has progressed to things like carditis or neuroborreliosis?
For those more severe cases, we often use intravenous antibiotics like ceftriaxone. They're powerful drugs that can reach those harder-to-reach areas and effectively fight the infection.
So we definitely have options, but I've heard that treating Lyme arthritis can be particularly challenging. What are your thoughts on that?
Yeah, you're absolutely right. Lyme arthritis can be a tough one to treat. Sometimes antibiotics alone aren't enough to get rid of the inflammation and pain. In those cases, we might consider adding anti-inflammatory medications or, in extreme cases, even exploring surgical options.
It's a good reminder that Lyme disease can have a lasting impact on patients' lives, even after the initial infection is gone.
I agree. It's a complex disease, and managing those long-term consequences can be a real challenge.
Okay, so let's switch gears for a moment and talk about relapsing fever, another interesting infection caused by Borrelia species.
Relapsing fever is kind of like a hidden gem in the world of Borrelia infections. It doesn't get as much attention as Lyme disease, but it's a really fascinating illness in its own right.
Okay, I'm intrigued. What makes relapsing fever so fascinating?
Well, as the name suggests, it's marked by these recurring episodes of high fever chills, headache, and muscle aches. But the truly fascinating part is the mechanism behind these relapses. It's a testament to Borrelia's incredible ability to outsmart our immune system.
I knew there had to be some clever microbial strategy going on. Tell me more.
Relapsing fever is caused by a few different Borrelia species, and it's transmitted either by ticks or lice. The interesting part is how these bacteria manage to avoid the immune system and cause those recurring fevers. It all boils down to their ability to change their identity to constantly reinvent themselves.
Hold on, they can change their identity. How's that even possible?
They have this remarkable ability to change their surface proteins, specifically a protein called variable major protein, or VMP. It's through a process called antigenic variation that Borrelia can switch out different versions of this VMP, essentially changing its appearance to the immune system. It's like they're wearing a different mask each time they encounter the immune system.
So the immune system attacks, clears the bacteria, thinks it's won the battle, and then boom, Borrelia switches its outfit and the whole cycle starts over. That's wild.
That's exactly how it works. It's like a never-ending game of cat and mouse between Borrelia and the immune system. So we were talking about how Borrelia can actually change its identity. How does that happen? It sounds almost too clever.
Right. It's like a sci-fi movie, bacteria shape-shifting to get away from the immune system.
Yeah.
Tell me how it works. How does this microbial magic trick actually happen?
Well, it all comes down to this one gene that makes a surface protein. It's called variable major protein, or VMP for short. This VMP is like a costume that Borrelia can change whenever it wants.
So this VMP helps Borrelia hide from the immune system.
It's actually more than just hiding. It's about constantly changing its appearance so the immune system can't keep up. This is what's called antigenic variation.
Okay, so break it down for me. How does this antigenic variation work in relapsing fever?
Okay, so imagine this. You've got this initial wave of Borrelia invading the bloodstream, causing that first high fever. The immune system kicks in, starts producing antibodies to target the bacteria's specific VMP. Everything seems to be going well. The bacteria are being cleared, the fever goes down. But just when the immune system thinks it's won, bam, Borrelia changes its VMP.
So those antibodies are no longer effective because the bacteria is basically wearing a new disguise.
You got it. The new Borrelia population with its brand new VMP multiplies really quickly, and that triggers another wave of fever. This cycle of fever then relapse can happen over and over again, which is why it's called relapsing fever.
It's like a never-ending game of hide and seek. But does this go on forever? Does Borrelia have an unlimited number of these VMP variations?
That's an interesting question. Actually, it doesn't. As Borrelia cycles through its different VMPs, it eventually starts repeating earlier types. And this repeating might actually be what saves the immune system. It finally recognizes something familiar, and it can actually eliminate the bacteria more effectively.
So it's like the bacteria's own trickery ends up backfiring on it.
You could say that. It's really fascinating to see this constant back and forth between pathogens and the immune system.
Speaking of the host, let's talk about how relapsing fever actually affects people. What are the key clinical features we should be looking for?
Well, relapsing fever can be pretty dramatic. It usually starts all of a sudden with a really high fever, often reaching 104 degrees Fahrenheit or even higher.
Oh, that's a really high fever. What other symptoms do you see with this intense fever?
Along with the fever, you see chills, drenching sweats, headache, muscle, and joint pain and weakness. It can look a lot like a severe case of the flu. Some patients also develop a rash, nausea, vomiting, abdominal pain, and confusion. And if you examine them, you might find an enlarged liver and spleen, as well as tenderness in the abdomen.
So it sounds like it can affect multiple systems and really knock people down.
Yeah, it really can. And then after about 3 to 6 days of this intense fever, the fever suddenly goes away. The patient might feel like they're getting better, but that's actually a trick.
Because we know that the fever's going to come back right.
Right. After about a week, the fever comes back full force, along with all those other symptoms. And this cycle can repeat multiple times, usually somewhere between 3 and 10 episodes.
Each relapse must really take a toll on the patient. Are there any long-term complications to worry about?
There are, unfortunately. Those repeated bouts of fever and inflammation can really damage the body. Some of the more serious complications include myocarditis, hepatitis, pneumonia, meningitis, and even hemorrhage. And louse-borne relapsing fever is usually more severe and has a higher risk of these complications.
That's a good reminder that even though relapsing fever isn't as common as Lyme disease, it can be pretty serious.
You're absolutely right. It's definitely not an infection to be taken lightly.
Okay, let's talk about treatment. Are the same antibiotics used for Lyme disease effective against relapsing fever?
Luckily, yes. Doxycycline and tetracycline are usually the first-line treatments, and they're very effective in most cases.
So we have the tools to treat it. But what about diagnosing relapsing fever? How do we actually catch those sneaky Borrelia in action?
The best way to diagnose relapsing fever is actually pretty simple. It's just by looking at the patient's blood under a microscope during a fever spike. We're looking for those characteristic spiral-shaped Borrelia.
So unlike Lyme disease, where Borrelia can be hard to find, they're actually easy to see in the blood during those fever spikes.
Exactly. We can either use a wet mount or a stained blood smear, and the bacteria are usually there in large numbers and easy to see.
That's good to know. It sounds like a fairly straightforward diagnostic process. Are there any serological tests for this, like we use for Lyme disease?
There are serological tests that can detect antibodies to relapsing fever Borrelia, but they're not as widely used or as reliable as the tests for Lyme disease. So microscopy is still the most reliable way to diagnose it for now.
Sometimes the simplest techniques are the best.
I agree. Sometimes keeping it simple is the way to go.
Speaking of simple, let's talk about managing these Borrelia infections in a practical sense. What are some important things to consider when we're monitoring patients who are being treated, especially for those more serious cases of Lyme disease?
Monitoring is really important, especially for patients with Lyme carditis or neuroborreliosis. For Lyme carditis, we use electrocardiograms or EKGs to monitor the heart rhythm and check for any abnormalities.
So EKGs are essential for seeing how the heart is functioning electrically and catching any problems caused by the infection.
Exactly. And for patients with neuroborreliosis, we need to do repeated cerebrospinal fluid analyses to keep an eye on the inflammation in the central nervous system.
So basically, we're checking to see if the treatment is working to bring down the inflammation and protect the nervous system.
Exactly. These monitoring tools let us see how the patient is doing, make any necessary changes to their treatment, and ultimately help them get the best possible outcome.
It's all about providing individualized care and making sure each patient gets what they need. It's not just about tests and treatments, though, right? Patient education is a big part of it, too.
Absolutely. It's so important to give patients the knowledge they need about Lyme disease. We need to make sure they understand the potential long-term effects even after they've been successfully treated and that they could get reinfected.
That's right. Lyme disease isn't a one-and-done kind of thing. People can get infected again if they get bitten by another infected tick. What are some other important things you talk to your patients about?
I always stress how important it is to keep being vigilant and keep using prevention strategies even after they've finished treatment. Avoiding tick-infested areas, wearing protective clothing, using insect repellents, all of those things are still important to prevent future infections.
It can feel like a broken record, but sometimes those messages need to be repeated for them to really sink in.
You're right. It's easy to let your guard down once you've been through Lyme disease, but the risk is still there. And it's not just the physical aspects of the disease either. I also try to talk to them about the psychological impact, which can sometimes be overlooked.
I agree. Sometimes that gets forgotten.
Dealing with a chronic illness, especially one like Lyme disease that can have lingering symptoms, can really impact mental health. So it's important to recognize the emotional side of things and encourage patients to get support if they need it.
I think that's a really important point. We need to treat the whole person, not just the infection.
Exactly. A holistic approach is so important for truly helping our patients deal with the complexities of Lyme disease.
Now, speaking of complex, let's talk about something that often causes a lot of debate and controversy in the world of Lyme disease, and that's chronic Lyme disease.
Ah, yes, chronic Lyme disease. That's definitely a complex issue with no easy answers.
So can you tell us what people mean when they say chronic Lyme disease, and why is it such a controversial topic?
Well, chronic Lyme disease is often used to describe symptoms that stick around for a long time, sometimes months or even years after the initial Lyme disease infection, even after standard antibiotic treatment. These symptoms can include things like fatigue pain and cognitive difficulties.
So it's basically those lingering symptoms that some patients experience even after the infection itself is gone.
Exactly. The reason it's so controversial is because there are different opinions about what actually causes these symptoms and the best way to treat them.
So some people believe that these persistent symptoms are caused by ongoing Borrelia infection, while others disagree.
Right. Some healthcare providers think that the symptoms are caused by Borrelia that's still hanging around even though standard tests might not detect it. And they often recommend long-term antibiotic treatment, sometimes for months or even years.
But a lot of other healthcare providers, especially infectious disease specialists, aren't so sure about this long-term antibiotic use, are they?
Yeah, they argue that there's no real scientific proof that these symptoms are caused by Borrelia that's still there. And they also point out that using antibiotics for a long time can have significant risks, like antibiotic resistance, allergic reactions, and messing up the balance of bacteria in the gut.
So it's a tough situation with strong opinions on both sides. What's your approach to managing patients who have these persistent symptoms, the ones who might fall into this chronic Lyme disease category?
I like to take a balanced approach and stick to what the evidence tells us. If there's no clear evidence that Borrelia is still active, I generally don't recommend using antibiotics for a long time. Instead, I focus on managing the individual symptoms and addressing any other underlying conditions that might be contributing to them. This might involve things like pain management, physical therapy, cognitive behavioral therapy, or treatment for other conditions they might have.
So it's about taking a holistic approach and focusing on improving the patient's overall well-being, not just trying to eliminate Borrelia.
You got it. And it's also important to really listen to the patient, validate their experiences, and give them support. These are often people who have been through a lot and they need compassionate care.
That's a great point. Sometimes just feeling heard and understood can make a huge difference for patients who are struggling with these difficult and frustrating symptoms.
I couldn't agree more.
All right, so before we wrap up our deep dive into Borrelia, let's test your knowledge with 5 challenging exam questions. Are you ready to see how much you've learned?
Hit me with them. I'm ready for the challenge.
Okay, great. First question. What is the main way that Lyme disease is transmitted in the United States? Now think carefully. This is a bit of a trick question.
I think I know this one. It's the Ixodes scapularis tick, also called the black-legged tick or deer tick.
That's right. Those little ticks are responsible for most of the Lyme disease cases in the northeastern and midwestern U.S. But don't forget that other Ixodes species can also spread Lyme disease in different parts of the country and the world.
Good point. The epidemiology of Lyme disease can change depending on where you are.
Exactly. All right, second question. Which Borrelia protein is really variable and can actually be used to predict how severe the disease might be?
That would be the OspC protein. It's a major factor in how the bacteria causes disease, and all its variations help Borrelia avoid the immune system and cause a whole range of symptoms. If we analyze the different OspC variations, we can get a better idea of how aggressive a particular strain might be.
You got it. OspC is a really useful tool for understanding how Lyme disease might progress. All right, third question. What are the 2 most common serological tests that we use to diagnose Lyme disease?
Those would be the ELISA, which stands for Enzyme-Linked Immunosorbent Assay, and the Western blot. ELISA is usually the first test we do, and if it comes back positive or borderline, we confirm it with a Western blot.
Perfect. But remember, those serological tests for Lyme disease aren't perfect. We need to be careful about how we interpret them and consider the patient's symptoms and other factors. False positives can definitely happen, especially early in the infection.
Right. We can't just rely on the lab results alone. We have to look at the whole picture.
Absolutely. Okay, fourth question. What is the recommended treatment for early localized Lyme disease, the kind that usually presents with just one erythema migrans rash?
For early localized Lyme disease, we can usually treat it effectively with oral antibiotics. Doxycycline and amoxicillin are the ones we typically prescribe, and they have really good cure rates when they're given early on in the infection.
You nailed it. Early treatment is key for Lyme disease. If we catch it early on, we can usually prevent it from progressing to those more serious complications. Okay, last question. What is the most characteristic clinical feature of relapsing fever?
Well, relapsing fever, just like the name suggests, is characterized by recurring high fevers. Patients have these episodes of fever that last for a few days, then they seem to recover, but then the fever comes back again. This cyclical pattern is a big clue that helps us diagnose relapsing fever.
Excellent. You've clearly mastered the world of Borrelia. Congratulations on acing all 5 questions.
Thanks. It's been a really good review. I'm amazed by how much there is to learn about these bacteria.
Me too.
Yeah.
And I think we've really just scratched the surface. There's still so much more to discover about Borrelia and the infections they cause.
Definitely. I'm excited to see what the future holds for research on Borrelia.
Well, on that note, I think it's time to move on to our final segment where we'll share some additional insights and talk about the broader implications of what we've learned about Borrelia. We've covered a lot in our deep dive into Borrelia, from their structure and how they evade the immune system to the complexities of Lyme disease and relapsing fever.
It's been really interesting exploring these tiny but powerful bacteria.
It really has. So let's take a step back and think about some of the bigger implications of what we've learned. What are some key takeaways for medical microbiologists?
Well, I think the most important thing is to stay vigilant when it comes to emerging infectious diseases. Borrelia species are a perfect example of how bacteria can adapt and evolve and find new ways to survive in different environments. As human populations grow and we interact more with wildlife, we're likely to encounter even more tick-borne pathogens like Borrelia.
That's a good point. It's not just about Borrelia. It's about the bigger picture of infectious diseases and the challenges we face as healthcare professionals.
Exactly. We need to be proactive when it comes to public health. We need to invest in research to understand these pathogens, better develop effective diagnostic tools and treatments, and put strategies in place to prevent and control their spread. And it's not just about human health either. These tick-borne infections can also affect animal populations, which reminds us that we're all interconnected.
It's a reminder that our health is closely tied to the health of the environment and the other species we share this planet with. What about the clinical side of things? What can we as medical microbiologists do to improve the care we provide for patients with Borrelia infections?
Well, I think the fact that Borrelia infections can present in so many different ways and have long-term consequences highlights the need for a multidisciplinary approach to healthcare. Managing these infections often involves collaboration between different specialists, like infectious disease physicians, neurologists, cardiologists, rheumatologists, and even mental health professionals.
It's about recognizing that these infections can affect many different organ systems and that we need a team approach to provide the best care for our patients.
Exactly. And it also shows how important it is to have clear communication and coordination between healthcare providers, researchers, and public health officials. We need to work together to share information, develop best practices, and stay ahead of these evolving threats.
It's about breaking down those silos and working together across different disciplines. And for us, as medical microbiologists, our role in all of this is absolutely essential.
Absolutely. We're the ones on the front lines identifying these pathogens, understanding how they work, and developing strategies to fight them. We play a key role in guiding diagnosis, treatment, and prevention efforts.
It's a challenging role, but also incredibly rewarding.
I couldn't agree more. And the more we learn about Borrelia species, the better equipped we'll be to protect the health of both people and animals.
Well, on that note, I think we've come to the end of our Borrelia deep dive. It's been an amazing journey exploring these fascinating and challenging bacteria.
It's been a pleasure joining you on this journey.
And to our listeners, thank you so much for joining us on this deep dive. We hope you found it informative, insightful, and maybe even a little bit inspiring. The world of microbiology is full of wonders and complexities, and we encourage you to keep exploring and learning. Until next time, happy microbe hunting.